What's up, beautiful people? Another day, another study. We've talked a lot about dementia, so we've got another here, dementia study. But one of the things I've talked about is how cognitive decline can take decades. And when we look at drug therapy, nothing has really impressed me as far as what we've got in that arena, likely because once someone is already diagnosed, there's been significant degradation in brain structure and also in brain function. We've talked about cognitive resilience and cognitive reserve and some of these concepts before. One of the things I'm really interested in is prognostication. So part of my business is doing what I call health risk assessments, doing a comprehensive in-depth analysis of biomarkers and risk factors and coming up with a plan to mitigate risks for chronic disease as much as possible. We know the worldwide dementia incidence is expected to surge to approximately 153 million people in 2050. We know that our current treatments for dementia are lacking. Therefore, what I really am interested in is how do we risk assess people? Are there ways that we can look at who is at risk for cognitive decline, figure out those factors, and then address them? So, this was a study looking at the microbiome-derived metabolites. I'm less interested in the composition of the microbiome because there's so much that we don't know. We know that a healthy microbiome is probably a little bit more diverse, contains certain bacterial species associated with things like short-chain fatty acids. Outside of that, a lot of the microbiome sphere is still the wild, wild west, and we still have a lot to learn. However, looking at what the microbiome makes, I think is very interesting because you can have people who have the same bacteria, that those bacteria have a different expression of the things that they make downstream. And so this study utilized what we call metabolomics, which is looking at those metabolites and looking at healthy controls, people with subjective cognitive impairment, and then people with a clinical condition, which is mild cognitive impairment, which is actually diagnosed with some pretty stringent clinical criteria. So when they looked at the differences between these groups, there really wasn't significant differences between the different microbiome components at the highest level, at the phylum level. At the genera level, which is a subset of the phylums, there was some differences between the groups in 10 different ones. When they looked at the metabolites, 5 metabolites were associated with early cognitive decline, and these were mostly in tryptophan related metabolic products. So tryptophan is amino acid made famous by turkey. Why did you get tired when you eat the turkey? It's not so much the tryptophan. It's more of you probably ate a lot and then sat around and did nothing, right? Anyway, so these 5 metabolites were choline, 5-HIAA, which is a serotonin metabolite, IPA, and those people who had lower concentrations of those, you saw that in the subjective cognitive impairment and the mild cognitive impairment compared to healthy controls. Indoxyl sulfate, which is a kidney and cardiovascular toxin, had increasing levels as you went from healthy to SCI to mild cognitive impairment. And then Kynurenic acid, which is actually a neuroprotective and antioxidant properties, was higher in the SCI and MCI controls. We see this sometimes in brain health where when you have injuries or insults or things like that, you get an increase in protective molecules, like in this case, the Kynurenic acid. However, when we make this, that pathway that goes into this metabolism has some not so favorable byproducts. So it can be a marker of injury, but it's also a surrogate marker of a abnormal, not so much abnormal, but a non-beneficial metabolic pathway if it's left upregulated for too long. So really interesting study. These results are totally exploratory. I wouldn't take these home. I wouldn't measure levels of these things. We still have a lot of research to go in this area, but I'm really, really excited that we're looking at these prognostication factors and trying to figure out how we can identify who's at risk, and then hopefully down the line, learn other ways that haven't been shown in clinical studies to decrease our dementia risk.